What if you could press the pause button on your own biological clock? Scientists have just successfully integrated tardigrade CAHS proteins into human cells, triggering a reversible state of molecular gelation that freezes metabolism in its tracks. For the first time, we are hijacking nature's most indestructible blueprint, the tardigrade, to stop cellular decay forever. This is not science fiction. This is the future of aging. Meet the tardigrade. Also known as the water bear, this microscopic animal measuring less than half a millimeter is the most resilient creature on Earth. It survives the vacuum of space, boiling temperatures, extreme radiation thousands of times beyond human tolerance, and decades without a single drop of water. When conditions turn lethal, it doesn't fight. It enters a state of suspended animation called biostasis, essentially pressing pause on its own existence until conditions improve. When a tardigrade dries out, it shrivels into a tiny ball called a tun, reducing its body volume by up to 95%. At this point, its metabolism drops to undetectable levels. It becomes a glass-like structure, neither alive nor dead, preserved in a state of suspended animation for decades. When water returns, the tun rehydrates and the tardigrade resumes full activity within hours, as if nothing happened. The secret to this feat lies inside its cells. Inside every tardigrade cell are unique proteins found nowhere else in nature. Cytoplasmic abundant heat-soluble proteins, or CAHS. These are intrinsically disordered proteins, meaning they have no fixed shape. They float freely in the cell until stress triggers them. When dehydration or osmotic shock hits, CAHS proteins self-assemble into a fibrous gel-like network that fills the entire cell, creating a protective molecular scaffold that stabilizes everything inside. This gelation process is remarkable. CAHS proteins form nanometer-wide fibers that cross-link into a three-dimensional matrix, essentially turning the cell's liquid interior into a solid gel. This molecular glassification locks proteins, membranes, and organelles in place, preventing them from unfolding, aggregating, or collapsing. Scientists have shown that this gel reduces molecular motion so effectively that it halts metabolic activity without causing permanent damage to the cell's machinery. In 2024, researchers at the University of Wyoming led by Sylvia Sanchez-Martinez and Thomas Boothby made a stunning discovery. They introduced tardigrade CAHS-D proteins into human cells. Amazingly, the proteins did the exact same thing they do in tardigrades. They formed fibrous gels and slowed down the human cell's metabolism. When stress was applied, the cells entered a reversible biostasis. When the stress was removed, the gels dissolved and cellular metabolism returned to normal. The implications are staggering. Human cells containing CAHS proteins survived osmotic shock significantly better than control cells. The gelation reduced metabolic activity, protected organelles, and maintained cell integrity. Importantly, the process was fully reversible. As Boothby put it, when the stress is relieved, the tardigrade gels dissolve and the human cells return to their normal metabolism. We have effectively transplanted the tardigrade superpower into our own biology. But CAHS is not the only weapon in the tardigrade arsenal. The damage suppressor protein, Dsup, binds directly to DNA and acts as a physical shield against radiation and oxidative damage. In 2025, researchers showed that Dsup protects yeast cells from oxidative DNA damage and extends their lifespan. Dsup wraps around chromosomes like a protective cloak, deflecting hydroxyl radicals that would otherwise shred our genetic code. This opens doors for cancer therapy protection and space travel. In 2026, scientists at the University of Michigan and University of Chicago proved that CHS12 protein could protect synthetic cells through complete dehydration and rehydration cycles. These synthetic cells retain the ability to read DNA and produce proteins even after being dried and restored for weeks. This breakthrough means we can now store complex biological materials without refrigeration, a game-changer for medicine delivery, vaccine distribution, and biomanufacturing in remote areas. Here is where aging comes into focus. The 2024 study showed that CHS-induced biostasis slows metabolism in human cells. Slowing metabolism correlates directly with reduced cellular aging. By transiently inducing this gel state, we can theoretically pause the accumulation of oxidative damage, protein misfolding, and other hallmarks of aging. Researchers believe this could lead to technologies that slow the aging process itself, buying us precious time against biological decay. The practical applications are already being explored. Imagine storing transplant organs for months without refrigeration. Imagine delivering temperature-sensitive vaccines to remote villages without a cold chain. Imagine astronauts suspended in biostasis during multi-year journeys to Mars. Imagine cancer patients whose healthy cells are temporarily gel-shielded during chemotherapy. All of these are now within reach, thanks to a protein borrowed from a microscopic animal. Tardigrades have survived 5 mass extinctions across 500 million years. Their secret was always hiding in plain sight, a set of proteins capable of freezing time itself. Now we have cracked that code. By bringing CHS proteins into human cells, we have learned to pause our own biology. The ability to slow aging, preserve life, and explore the stars may all trace back to this tiny water bear. The question is no longer if we can press pause on aging, but what we will do with the time we gain.